Hello, everyone. A very good afternoon to all of you. How are you all doing? Great. Okay, so welcome to Swiflearn online classes. As you already know, that Swiflearn is the India's most sincere learning destination from classes 1 to 10. Over here, all the classes are 100% live and interactive classes. Like if you have any sort of doubt, you can ask to the teacher in the classes only, and the teacher will explain everything in the classes. Right. Coming to the second thing, we give personalized attention to each and every students of our batches because maximum 6 students are there in each of our batches. The batches can be of 1 student, 2 student, as per you will select your batch. So maximum 6 students are there, and this basically help us to keep an eye on you. And also, without any hesitation, you can ask any sort of doubt regarding the topic in the classes. Right. Coming to the third thing, that is, we have a very good dedicated academic mentor team who continuously track how you are progressing. So if you are lagging in any sort of topic, so definitely you will get the feedback, and this feedback will help you to improve yourself, right? And coming to the fourth and the most important thing, that is, we cover the syllabus as per your school examination schedule. So if you have some unit test or any sort of test coming in your school, and the topic which are coming, but we have not done it yet in Swiflearn, so we will go with those topics first, and then we will move on to the rest of the topics, right? So like this, we generally handle the custom request. So apart from that, we have a lot of different doubt classes, PTM and all. So let us see what are our key features and why this Swiflearn is totally different from any other platform. First of all, we maintain a detailed progress report of each and every students, and with the progress report there will be the PTM also. So जैसे आपके स्कूल में भी PTM होता है, इधर भी PTM होता है, and here your parents will be able to directly interact with us, and they can give us the feedback. They can share many experiences with us. Okay, so we are always here to hear from you. Now, coming to the second thing, that is the Swiflearn Academic Test Series, in short, which we call as the SAT. So, this happen in every Sunday from 9 a.m. to 5 p.m. You can perform over there, and your name and the rank will be given on our leaderboard. Fine. Coming to the third and the fourth most important thing, that is the practice. You already know that the practice makes a man perfect, right? So, what you have to do is, after completing each and every chapter from your textbook and your revision notes, what you have to do is you can solve a lot of different question and answers. So we have unlimited practice test, both subjective and objectives. So objective question answer you can solve it, and subjective questions, that is very very important, you can write your answer and submit it along with the diagrams, chemical equation, whatever is needed for the particular type of question. You can write it and submit it so that we can check your answers, and along with checking the answers, we will give you the tips and tricks how to improve your answer and how you will get the full marks by writing the proper answer according to the marks, right? So this is very very important for you to get the full marks. And you, as you already know, that in maths and sciences, it's really very easy to get the full marks in board exam also, right? Okay, so for experiencing all these things, what you have to do is you have to book a free trial class. So all the trial classes are free of cost. Book it anytime whenever you are free, and then enroll your name in our regular batches. Okay, and we will meet every day in the regular batches. ठीक है ना? Chalo. So let's now dive into our topic of today. And before that, let me tell to those who have not subscribed our channel, please subscribe it and press the bell icon so that you will never miss any of the update of the upcoming sessions. And do not forget to like, comment, and share also. So, today, as you already know, that we are going to start with a new topic, that is the structure of chromosome, cell, and cell division. So, this is the chapter which is there in class 10th syllabus of ICSE board. So this is very very important because you are now studying the basics of the genetics. So the whole genetics is dependent upon the chromosomes, the cell division, and after that many different things will come. So the cell, that is the basic unit of life, and how cell divides, and what are the basic things which are present within the cell that are giving all the characteristic features to our body. This is very very important. So we need to know about all these things. First of all, we will study about the chromosomes, then the discovery of the chromosome, then we will come to the chromatin. So there is a very huge confusion between the chromatin and the chromosome. So it happens to all the students. So be very careful. Chromosomes and chromatins, what are the differences between them? I will discuss this with you in this session. Next, we will move on to the structure of the chromosome, then the genes. As you already know, the hereditary characters are basically carried by the genes from the parents to the offspring. So what are these genes? Yes. And then we will move on to the need for the new cells, the cell cycle, cell division, the different types of cell division, mitosis and meiosis. And in the mitosis, we will see about the karyokinesis and the cytokinesis. And then finally, we will talk about the meiosis. So these are the different things which we are going to discuss in today's session. Okay, all of you are ready? Yeah? Okay. So let's start with the introduction of the chromosome. So what are chromosome? As I have already got an idea that you already know the word chromosome, and you already know that this is the thing which is present within the nucleus. Whether it is a plant cell or an animal cell, all the cells are having some chromosomes within that. That is the thread-like structure. Okay, now when we will talk about the chromosome, then we can see only through the microscopes, and they are only seen during the cell division. And when the cell is not in the position of dividing, then we will see a very complicated thread-like structure, bits in a thread-like structure, which is known as the chromatin. So we will talk about the chromosome and the chromatin. So chromosome is the condensed structure, and when we will unbind it, we will get the chromatin. ठीक है. Now ये chromosome क्या होता है? Pairs में रहता है always. After the replication of the DNA, means the DNA duplication or replication, हम दोनों ही बोल सकते हैं. तो डुप्लीकेट होने के बाद क्या होता है? The 23 will become a 23 pairs, and these pairs are nothing but the chromosomes within the nucleus. So before going to the detail of the structure and function, let us see a video of the chromosome.
Chromosomes. In the nucleus of each eukaryotic cell, the DNA is packed together into chromosomes. Each chromosome is made up of DNA that is tightly coiled around proteins that give it the structure. This tight packaging system means that 2 meters worth of human DNA can fit into a tiny cell that is only 2 micrometers wide. In this video, we are going to look at what chromosomes are and what their role is. Chromosomes usually occur in pairs, except for gametes, which are eggs and sperm cells, but the number of chromosomes changes depending upon species. Humans have 23 pairs, elephants have 28 pairs, and carrot plants have 9 pairs. If you want to learn more about DNA, watch this video. So chromosomes are made from long, coiled molecules of DNA known as double helix. Within this long chain, there are shorter regions that carry the genetic code for particular proteins that are known as genes. Chromosomes carry all of the information that help a cell grow, survive, and reproduce. Each chromosome has a centromere, which divides the chromosome into 2 sections, or arms. The location of the centromere on each chromosome is what gives it its characteristic shape. The P arm are the short arms, and the Q arms are the long arm structure. Chromosomes are passed from parents to offspring and play an important role that ensures DNA is copied and distributed accurately in the process of cell division. Chromosomes are not usually visible in the cell's nucleus and can only be seen when the cell is dividing. During cell division, the chromosomes are duplicated. We will look at this in more detail in future videos, such as what is mitosis. When the chromosomes are duplicated, they condense into short structures which can then be strained and observed under a microscope. The centromere holds the duplicated chromosomes together. Duplicated chromosomes are commonly called sister chromatids. So, from this video, you should know that chromosomes hold the DNA inside the nucleus. They have a centromere, which holds the duplicate chromosomes together, and these duplicates are known as chromatids. Humans have 23 pairs of chromosomes, but other species have different amounts.
Yes, so from here we have already got an idea about the chromosomes, right? So now where these chromosomes are present, these chromosomes are found within the cell. Now, what within the cell? Within the cell, we already know that in eukaryotic cell, we have a proper true nucleus. And within the nucleus, these chromosomes are present. Just see, look at the picture of the chromosome. This is the basic structure of the chromosome. You can see the different arms. These are the arms of the chromosome. And all the arms are joined at the center, which is known as the centromere. Okay, so these arms of the chromosomes are also known as the chromatid. ओके तो क्रोमैटिन अलग है क्रोमैटिड अलग है क्रोमोसोम अलग है ठीक है क्रोमोसोम के अंदर जो भी आर्म्स होता है वी कैन कॉल इट एज द क्रोमैटिड एंड व्हेन दे आर शेयरिंग अ सिमिलर centromere we will call them as the sister chromatid. I guess you have already heard about the name of the sister chromatid, right? Now let's now see who first discovered this chromosome. So while doing some plant experiments or the examining the different plant cells, a botanist, very famous botanist, Carl Wilhelm von Nageli in 1842, he discovered the chromosomes. And then he gave the structure of the chromosome. He saw that it is the chromosome which are X-shaped structure which are present within the nucleus. So you can see over here, this is the X-shaped structure. The end of the chromosomes are known as the telomere. This is the centromere. And if you unbind this chromosome, you will get the DNA. Okay, so if you are asked like DNA is different, chromosome is different, no, the DNA structure, the DNA double helical structure is bind and condensed to form the chromosome. Okay, all of them are present within the nucleus. Now what about the chromatin then? Chromatin is nothing but when the chromosomes are unbinded and they are not in pair, they are unpaired, and when they can be seen only under the electron microscope and not during the cell division. So from here we can get a lot of different types of differences or the variant like varieties of the chromatin and the chromosome. So chromosomes are the paired structure which can only be seen during the cell division, but chromatin are the thread-like structures which can be seen at any stage of the cell cycle other than the cell division stage. Okay, so they prevent the DNA damage, they support the DNA to permit the process of the cell cycle in mitosis and meiosis. So let's see how we can unbind them and we can get the chromatin fiber. So you can see the X-shaped structure, the condensed structure of the chromosome. Now when we will get this thread-like structure from it, this thread is known as the chromatin fiber. So we can see this chromatin when the cell is not in the stage of division. Then if we again see them under the microscope and unbind it, we will see the different histone proteins and the DNA double helical structure. So the histone proteins and the DNA double helical structures are together present within the nucleus and forming the most condensed structure, which is the chromosome. Up to this much clear hai? Now let's see what are the differences between the chromosome and chromatin. If you get this type of question, you will have to write like this, making the different columns as you already do in biology. So here is the chromosome. These are the condensed structure, but in case of the chromatin, they are the thread-like structure. And in this thread-like structure, the histone protein and the DNA structures are present. Okay, second is chromosomes are always in pair. So if it is in our cell, that will be 23 pairs, and if it is any other animal cells, it will be dependent upon the different species. And chromatin is unpaired, they never remain in pair. In case of chromosome, we can see them only during the cell division, but in case of chromatin, we can see them any other stage other than the cell division stage. Okay, and chromosome is highly visible under the microscope, that is light microscope or compound microscope, because they are the condensed structure, we can see them easily. But in case of chromatin, we can see them only under the electron microscope, only under very high power. Up to this much clear hai? Yes. Now see, now let's move on to the structure of the chromatin. So we already got an idea about the structure of the chromosome, that is the X-shaped structure. Now if I just unbind it, we will get the chromatin, which contain the DNA and the histones. So histone is one type of protein. What is DNA? Yes, can anybody tell me what is the full form of DNA? Yes. Deoxyribonucleic acid, right? Very good. So, this is the single molecule of DNA, that is the deoxyribonucleic acid, which contain the different hereditary characters from the parents to the offspring. So, these are passed from the parents to the offspring, and that is why we sometimes look a little bit similar to our father or mother. We get the different hair color, or hair structure, or eyeball color, or different types of characteristic features which we get from our parents, right? So this DNA contain the direction and give you the uniqueness. Whatever creature is there, like whether it is a plant or an animal, the DNA which are present within them, they will give them the unique characteristic feature. Now let us see what is the DNA with the help of another video.
In this video, we are going to learn about DNA, where it is found, and what it actually is, and what it is made of. All living things have DNA, or in the case of some viruses, they have RNA instead. DNA stands for deoxyribonucleic acid. Let's start with where it is found. Most cells are eukaryotic. Animal cells, plant cells, and fungi, etc. Inside eukaryotic cells, you have a nucleus. Within most nuclei, there are X-shaped chromosomes. Chromosomes occur in pairs, but the number of chromosomes change depending upon the species. Humans have 23 pairs, elephants have 28 pairs, and carrots have 9 pairs. DNA is found within these chromosomes. Archaea and bacteria are prokaryotes, and because they do not have nuclei in their DNA, it is located in their cytoplasm. So what is DNA? It's a large, complex molecule that carries the genetic information that determines the characteristics of living things. And other than identical twins, we all have our own unique DNA. DNA contains lots and lots of sequences. Some sections of these sequences have a specific genetic code for making particular proteins. And these special sections are called genes. A gene is just a short section of DNA, and because each gene codes for a specific protein, it is in these genes that give us our individual characteristics, such as whether you have blue eyes or brown eyes. Proteins are the building blocks of life, and as our genes code for specific proteins, this is how DNA controls our body and helps us to survive, develop, and reproduce. But what is DNA made up of? DNA is made up of lots of molecules called nucleotides. DNA is a polymer because it is a large molecule composed of repeated subunits, the nucleotides. Each nucleotide contains a phosphate group, a sugar group, and a nitrogen base. When people think of DNA, they usually think of these bases. There are 4 bases: adenosine, thymine, cytosine, and guanine. But just remember A, T, C, and G. And it is best to remember in this order: A, T, C, G. Because A and T bases pair up, and C and G bases pair up. The bases pair up, forming hydrogen bonds, and the DNA forms a twisted shape that is known as a double helix. So there you have DNA. You should know that it is usually found in the chromosomes, within the nuclei of cells. It is a polymer, and it is made up of lots of nucleotides that contain bases. There are 4 bases: A, T, C, and G. and the bases pair up as AT and CG.
Yes, so at least you have got an idea how the DNA looks like and what are the different chemical compositions of DNA, right? So let's see who discovered the DNA first. So the scientist Johannes Friedrich Miescher in 1869 first was doing the research on the white blood cells, that is the WBC, and first identified the DNA. So a lot of different types of experiments were going on, and there were a lot of different shortcomings that DNA is the genetic material or not, and finally it is proved and it is believed to be the genetic material, and it carries all the different types of genes from the parents to the offspring, even like in the human being, in the different other animals, as well as in all the different plants. So let us now see what are basically the chemical composition of the DNA. So as you have already seen in the video that the DNA consists of the sugar, the pentose sugar, the phosphate backbone, as well as the four nitrogenous bases. So what are the four nitrogenous bases? The four nitrogenous bases are adenine, thymine, cytosine, and guanine. So adenine and guanine will come under the purine, and thymine and cytosine will come under the pyrimidine. If you see, you will observe that A and T always pair with each other and C and G always pair with each other. So when they pair up, they basically are bound by the hydrogen bond. So in between the A and T, there will be two hydrogen bonds, and in between the C and G, there will be the three hydrogen bond. This is totally fixed in any sort of DNA. Okay? Up to this much clear? Now, for this, the Chargaff, the biologist or biochemist, Arvind Chargaff discovered and gave a theory that is known as the Chargaff's rule. He said and first discovered that the number of A, that is the adenine, is always equal to the number of T, that is thymine, because the A is only pairing with the T, so definitely their numbers will be equal, as well as C, that is the cytosine, is always equal to the number of the guanine, that is the number of C and G are equal to each other because they are binding with each other. Okay, and when they are binding, basically they are binding with the different hydrogen bonds. So there are two hydrogen bonds in between the adenine and thymine, and there are three hydrogen bonds in between the cytosine and guanine. This is known as the Chargaff's rule. Clear hai? Yes. Now, other than this, other than the DNA, there are the histone proteins, and the DNA is binding like this, the histone proteins, and giving it a proper stable structure, because the histone proteins are positively charged and DNA molecules are negatively charged structure. Together, they will make the whole nucleosome as a neutral structure. Okay, so this is the total nucleosome. Let me show you the structure of the nucleosome. You can see this is the DNA. These are the DNA which is binding the histone proteins. These yellow colored molecules are the histone proteins. And when this histone protein is totally bind by the DNA, this is known as the nucleosome. Okay, and finally when they are condensed, they will form the structure of the chromosome. So let us see the 3D model of this chromosome structure. Okay, so you can see this is the X-shaped structure which is known as the chromosome. The middle of the chromosome, as I have already mentioned, it is known as the centromere, where the different arms are joining with each other. This is the Q arm, that is one arm of the chromosome, and another arm is the P arm, the short arm. The shorter arm is known as the P arm. Then if you just unbind it, we will get the chromosome fiber, or we can say call it as the chromatin. Then we will see this type of structure where the DNA and the histone proteins are bind with each other, and they form the 6 molecular structure which is known as the solenoid. Again, after unbind it, we will get the most simple structure which is known as the nucleosome. You can see this yellow colored stone like structures are the histone proteins, and this blue thread like structure is the DNA which is binding them. Okay, and finally you can see this is the histone, and finally you can see the double helical structure of the DNA. Up to this much clear hai? Yes? Up to this much got it? Chalo. Let's now move on to the questions. Let's revise some of the facts what we have done. Anybody tell me what is the full form of DNA? Yes, quickly. It is nothing but the deoxyribonucleic acid, right? And what is Chargaff's rule? Yes. So Erwin Chargaff proved that the number of A, that is the adenine, is always equal to number of T, that is thymine, and they are binding with each other with 2 hydrogen bonds. And C, the number of C will be always equal to the number of guanine, that is G, and they are binding with each other through 3 hydrogen bonds. These are coming under the Chargaff's rule. Up to this much totally clear hai? Yes. Now let's move on to the gene. So as you already have an idea, like the genes, they are basically giving you the different characteristic features. So what are genes? These genes are specific region of the DNA which is giving a proper coding to the proteins. Okay, and every person have 2 copies of each of the genes, one getting from the father and another one getting from the mother, right? Now you can see the position of the genes. You can see this is the cell, within it a nucleus is there, within the nucleus the chromosomes are there, and within the chromosome it is the DNA. We already know that the condensed form of the DNA and the histone proteins are making the chromosomes, and only the structure which can code for a particular protein, those structure is known as the gene on the DNA, which are giving a characteristic feature to our body. Okay, now let's see how they are going from the father and mother to the next generation. So definitely they will go through the cell division processes, and also the genetic materials will go from one cell to another through the cell division process. So there are 2 main types of cell division. One is the mitosis and another one is the meiosis. So what is the main need, or yes, the main purpose of the cell division or getting a new cell? First of all, it is very important for growth and development. So you know the babies, after just after birth, their size is very small, but they grow. Why? Because of the continuous cell division within their body. So it helps in the growth and development. It helps in the replacement as well as the repair. So let us see one by one how it happens. So you can see the cell is dividing again and again, and it is increasing size and increasing mass also, and also increasing in number, so that the total net volume of the body will also grow. Secondly, for the replacement, you can see that the old cells are replaced by the new cells. So this replacement of the cell is very, very important inside our body. So there are the different types of lifespan for the particular types of cell. If you think about the sperm cell, they have a lifespan of only 3 days. If you think about the brain cells of the neuron, they can stay for a lifetime, and generally the cell have a lifespan of 7 years. Okay, and the third one is for repairment. If you have any damaged tissues, if you have a cut on your head, or if you think about the lizards, if the tail is cut, so what happened after few days, you will be able to see that again a new tail is grown. So this happened due to the regeneration or repairment of the cell and rebuilding the damaged or the lost structure. Okay, up to this much clear? Now let's move on to the cell cycle. So what do you mean by cell cycle? This is nothing but the series of event which is starting from the birth of the new cell till the next division. So we already know that from the mother cell, the 2 daughter cells are coming out. So from starting of the 2 daughter cell till the next division, when they will mature and again cell division will occur, the whole cycle is known as the cell cycle. So you can see it is starting from the G1 phase, that is the growth phase, then the synthetic phase, or the where the DNA duplication will take place, and after duplication they will again grow and become mature so that they can divide, and the cell division phase is known as the mitotic phase. Okay, Other than mitotic phase, the G1, G2 and S will come under the interphase. Together they are known as the interphase, and this is the mitotic phase where the cell division will take place. Again, if you can divide this mitotic phase, we will get one karyokinesis, the first phase, and second is the cytokinesis. Karyokinesis is what? That is nothing but the division of the nucleus. You can see over here, first the nucleus is dividing and then the cytoplasm is dividing. The karyokinesis is nothing but the division of the nucleus. You can see it will go through the four phases, the prophase, metaphase, anaphase and telophase. Okay, so these four phases, through these four phases the cell will go, and finally the nucleus will be divided into two. Yep. And after the division of the nucleus, the cytoplasm will divide, and finally they will detach from each other and the two new daughter cells will be formed. So this is the two different phases of the mitosis. Now let's move on to another type of division, that is the meiosis. So always remember, the meiosis takes place within the gametes only. If we are talking about the human body, then in case of the male it will happen within the testis, and in case of the female it will happen within the ovary for the formation of the male and female gametes, that is the sperms and the ovum. Okay, now here they never occur in pair, rather they will divide twice, and the amount of genetic information or the amount of chromosome will be totally reduced to half. Okay, and they will divide twice to form the four daughter cells. This is all about the meiosis. Now what is the significance of meiosis? First of all, the chromosome number is becoming half, so that again while the fertilization will take place, that is the sperm and the ovum will fuse with each other, they will again restore the normal number of the cell, sorry, normal number of the chromosome. Okay, so 23 is coming from the father and 23 coming from the mother. Together again it will become 23 pairs, or we can say 46 chromosome in the zygote. Okay, and another thing is there, the mixing up of the gene is very important. The mutation, or we can say the cross joining, when the paternal and the maternal chromosomes are separating, sometimes they come in contact with each other and they just exchange their um, codes of the DNA. Okay, they just exchange their DNA and form the varieties of the characteristic feature. So this recombination and formation of the variation is very important for our survival. Okay, so that is why this meiosis is very important. Let's now see some of the question answers, like why meiosis is referred to the reductional division. As I have already mentioned that while meiosis takes place in case of this type of cell division, the number of chromosome is reduced to half, that is from 2n, or the diploid number of chromosome, it is becoming haploid. Okay, only n. So that is why we can call it as the reductional division. So this question is very important. For 2 marks it can come in your exam. Okay, note it down. Now, let me tell you some of the facts about this DNA. So, throughout the evolution, the human have lost over 500 DNA codes. Can you imagine? Yes, and we share 40 to 50% of the identical DNA with a cabbage. And 9% of our DNA is the same to that of the other human being. Other percentages are all new or different from each other. Yes. So let's see what we have already covered in this session. We have studied about the chromosome, the discovery of the chromosome, the chromatin material, the structure of the chromosome, gene. Um, we already talked about the cell division, the need for the cell division, what is cell cycle, and cytokinesis, karyokinesis, and the mitosis and the meiosis. Right, so these are some of your extra time question. Try to solve this answer on your own so that this will help you in this board exam as well as the school exam. And thank you very much for staying till the end of the session. We will again meet in the next session. So before going, let me again remind you to subscribe our channel and press the bell icon so that you will never miss any of the update of the upcoming sessions. We will again meet in the next session, and do not forget to like, comment and share. Okay, so thanks a lot for staying till the end. We will again meet in the next session. So till then, bye bye, take care, stay safe, stay healthy.